all of you to the virtual launch of the Norwich Institute for Sustainable Development. This is a new center of excellence being set up by partners across the Norwich Research Park to improve global food security. So before introducing the institute itself, I wanted to start with a couple of thank yous and acknowledging all our partners. And I'm very happy that most of you are on the blackboard on the launch event today. I'm sorry, it's all virtual. We couldn't do it face to face. But first of all, I'd like to start with the John Innes Foundation for their support, which has actually helped convert this idea into a reality. And of course, all the partners, Dale Sanders of the John Innes Center, Ian Charles from the Quadrum Institute, Neil Hall from Earlham Institute, Nick Talbot and Jonathan Jones from the Sainsbury Laboratory, and of course, Laura from DEV, who have been very instrumental in bringing together this partnership across UEA and the Norwich Research Park. I also wanted to thank actually members of our core steering group, John McDonough and Heike from DEV, Konstantinos from the Norwich Business School, and Jonathan Jones, Kathy Martin, and Jacob Malone from the TSL and the John Innes Center. And I think even before, I feel very honored to be the director of this new institute, but even before I got into this role, I think this steering group and the leaders of these partner organizations have spent several years actually trying to work through and bring today to the point that we are actually launching this institute. So I would like to thank you all very much, and I hope that once things improve, we will actually be able to have a real get-together to celebrate over a glass of wine as well. So I think in the core team of the Institute, and who have been putting in a lot of work over the last couple of months, is Professor Aaron Verskool from DEV and Christopher Darby from the John Innes Center, who've really been very instrumental in pushing this forward. And recruiting now, we have our two full-time John Innes Fellows, Natasha Grist, who many of you will remember from DEV many years ago, and Peter Emmrich, who also is finished his PhD from the John Innes Center, and you'll hear more about Peter's work today after this initial inaugural launch event. So this institute is really, we're very pleased to be launching it now. It's a very timely initiative because especially in light of the COVID-19 pandemic, food insecurity has become one of the biggest global challenges today. Of course, also in the context of climate change, climate variability, and the very urgent climate crisis that we are confronting. So the Institute is really focused on finding solutions through engaging with transdisciplinary research. And we truly believe in this. It's not just adding a bit of social science or a bit of something and mixing with science, but it's truly working together in terms of finding solutions to conduct transdisciplinary research across disciplines, but also, I think very importantly, our approach, we are hoping, will be very inclusive. We'll look at different stakeholders, take different viewpoints, different approaches, different debates on board, not dismiss any point of view outright, but rather respect, built on a foundation of respecting different positions. And we hope that this will help us actually to co-produce knowledge, which accounts for difference, which accounts for diversity, which accounts for intersectionality, and will end up empowering the various participants with whom we are working, whether they are researchers, whether they are communities, whether they are farmers, whether they are NGOs or scientists. I think we want to empower each and every one of the stakeholders with whom we work. So without further ado, I'll request Natasha, we have a short film which introduces the Institute. So rather than it getting repetitive for you, it's just 2 minutes. We would like to play this launch video. Please forgive any awkward angles and positions. This video had to be shot entirely online, given the current situation in which we are. But that's the world we live in today.
Jonathan Jones met could not be read.
I would also request that can everybody mute their mics, so except when you speak, so we don't have background noise. And I will introduce Peter's seminar once we have seen the video. Perhaps the greatest challenge we face in the world today is how to feed and nourish our growing global population. We need to achieve this in the face of a grave climate crisis, whilst at the same time regenerating the natural environment that supports us all. This is a challenge no branch of science can address by itself. The Norwich Institute for Sustainable Development is our new approach to transform research and practice to create equitable, food secure, and sustainable futures. Together with our many partners worldwide, working with rural communities in our efforts to meet the Sustainable Development Goals.
For more than 100 years, the John Innes Foundation has been supporting plant science. This year, we're taking a particularly exciting step by backing this new collaboration between the John Innes Centre, UEA, and the wider Norwich Research Campus.
The University of East Anglia is well known internationally for its research excellence in climate science and international development. We've always believed in doing different, and the realisation of a sustainable world requires bold and imaginative action inspired by a research-based vision. This new collaboration is an important step towards envisioning a sustainable future through bridging disciplinary boundaries and uniting the brightest minds.
At the heart of the John Innes Centre is world-leading research into the fundamentals of plant microbial science. We balance this with translating our findings into innovations for the benefit of society, leveraging this research to address food, health, and environmental challenges around the globe. The Norwich Institute for Sustainable Development enables our researchers to work together with world-leading social scientists who maximise impact.
At the Norwich Institute for Sustainable Development, we are developing agricultural systems that are adapted to and mitigate climate change. With social scientists, agronomists, biologists, and environmental scientists working together, we can use the foresight that climate modeling provides to prevent global hunger in the decades to come. Thank you so much for that, Natasha, and thanks all for your patience. So I'm delighted now to introduce and welcome Peter Emmrich, who's a John Innes Fellow with the NISD, to present our first seminar today. Peter is going to be speaking on food security solutions, making the staple grass pea less toxic and improving farmer trust and uptake. Peter did his, as I said, his PhD at the John Innes Centre, and in his work he identified genes involved in the production of a toxin in the grass pea, which actually leads to severe problems, especially for communities who are already malnourished. And he has worked on how do you reduce this toxin from the grass pea in order to improve both food and nutrition security amongst the groups or areas where grass pea is endemic, which is mostly dry lands. So while Peter shares his presentation, just two requests to everybody. One is if you could mute your microphones while Peter is speaking. And secondly, if you have comments and questions, do feel free to type them into the chat box as Peter speaks. And depending on the time available for questions, we will ask some of you to speak your questions. But if there's not enough time, then we might combine the questions and take a few responses to that. So Peter.
Good afternoon. Can you see my slides and can you hear me?
Yes, your slides are now up full screen.
Excellent. A couple of years ago, I met a farmer in his field along a road in central Ethiopia. 3 months before that, he had planted chickpeas. It had been near the end of the rainy season, and he had hoped for a few more good showers. But no more rain fell, and in the weeks after, almost none of his seeds germinated. He became one of millions whose livelihood was threatened by drought. So he decided to use a tool that people in Ethiopia have been using for centuries to survive in times like this. He sowed a crop called grass pea, a pulse crop more resilient to drought as well as flooding than any other pulse. And the rain still did not come. But the grass pea germinated and grew on just that little bit of moisture left in the soil. 3 months on, this entire field of grass peas has never seen rain. This is why I work on grass pea. This is a crop we've been cultivating for 8,000 years now, and across Eurasia, North and East Africa. But one that is now widely grown only in Ethiopia and Eritrea and in South Asia, along with small pockets dotted all around the world in many other countries. This is what we call an orphan crop. But as the climate crisis progresses over the course of this century, the conditions faced by that farmer in Ethiopia will become more familiar to all of us. All around the world, rainfall and temperature patterns are shifting and becoming less predictable. Nutritious crops like grass pea that can survive a wide range of environmental stress can play a huge role in securing food supplies and farmer livelihoods. But there is a reason why grass pea is not much more widely used today. And it's because it produces a toxin called beta-ODAP, this compound here. If you are malnourished and eat grass pea as a staple food for several months, this toxin puts you at risk of becoming paralyzed. This disease has stoked historic food crises wherever grass pea has been a major food source. But when we say grass pea is an orphan crop, we mean that it both has received too little research attention, so few good varieties are available, and it is not grown and traded on a large scale, meaning there's little financial interest in improving it. And these two issues reinforce each other. In order to make use of all the potential that grass pea has to enhance food security, we have to address both sides of this. We have to remove the threat of toxicity and make sure breeders can easily produce new varieties. This is an issue for crop science. And we have to make sure that these new varieties are suited to farmers' needs and consumer preferences and support local food systems. This requires us to look at the issue through a social sciences lens. In this seminar, I want to talk about how we are combining both these approaches in the partnership of institutes that is now forming the NISD, and how this project fits with our broader vision. I'm happy that we have a very diverse audience here today, so please bear with me on the parts that you are already very familiar with. Starting with the issue of grass pea toxicity, the first question we had to answer was whether we could already find grass pea plants that produced none or almost none of the toxin. This was the first issue I tackled during my PhD. This included looking at the international diversity of grass pea landraces and varieties, and at a collection of grass pea mutants. Together with an Ethiopian colleague, I analyzed a collection of grass pea landraces from across Ethiopia. But with the exception of one line with somewhat lower levels, there was not much variation in ODAP levels. I also checked whether there was any geographic pattern in ODAP levels depending on where these grass pea lines were originally collected. But there too, there's no clear picture that would line up with altitude, temperature, or rainfall patterns. So I instead looked at international collections with grass pea lines from all around the world. Here there's clearly a broader spread of variation. One line came up with low ODAP concentrations, but this is a variety that had already been described and has already been used in breeding programs. But we know that certain environmental conditions can cause the plant to increase the concentration of ODAP in its seeds and leaves. And we want to make absolutely sure that there's no risk of toxicity, no matter what environment these plants are grown in. So having multiple parents with low ODAP traits that are caused by different genes allows us to pick and combine them to achieve reliably safe varieties. One way to broaden the pool of the available traits to breed crops is to use mutagenized populations. This is a well-established breeding technique that has been used for a century now, and which is considered a conventional breeding method. This involves treating a batch of seeds with radiation, or in our case, a chemical called EMS, to cause random mutations all around the genome. So we took this population of mutagenized grass pea plants and screened 37,000 of them for any that contained less of the toxin. That took a while. But I was able to identify 14 mutant families with reduced beta-ODAP concentrations, including some with lower levels than any varieties currently cultivated. Having several of these gives us a lot of material to breed with and to make sure ODAP stays low under any environmental conditions. Working in Nairobi, I've been crossing some of these low ODAP traits into accessions provided by our Ethiopian collaborators. This will allow us to develop varieties that are low in ODAP and also adapted to the specific environmental conditions in Ethiopia, and to maintain the diversity of grass pea in that country. This work is now also being replicated in Ethiopia and Morocco. At the same time, our collaborators are measuring the performance of low ODAP mutants along with their high ODAP relatives under stress conditions. We want to make sure that our new low ODAP lines are not compromised in any way. So far, we have no evidence that having much less ODAP harms the plant. But while there are many hypotheses, we still don't have a clear idea of what the plant gains from producing all this ODAP. By doing this research with our low ODAP mutant lines, we can also make sure that environmental stress does not raise the ODAP content to unsafe levels. In Kenya, we are investigating the use of grass pea as an intercrop with East African forage grasses. Like most legumes, grass pea can work with bacteria in the soil to fix nitrogen from the air into forms that are chemically available to the plant. This allows legumes to enhance soils, grow with very little fertilizer, and produce a lot of protein. By using grass pea together with a highly productive perennial grass, we hope this will improve soil health and enhance the sustainability of livestock feed in Kenya and beyond. But let's return to ODAP. We still didn't have a grass pea line that is at zero ODAP. But a bigger problem is that we still don't know where the genes that control ODAP production sit in the genome. This makes it very difficult for breeders to track this trait through their breeding processes. If we could find the genes that make the enzymes responsible for producing ODAP, then we could potentially produce zero ODAP lines by disrupting those genes. And we could make this trait very easy to track and test for. This approach, where we first identify a gene and then use it to get to the trait we want, is called reverse genetics. But to enable this, we first need a genome. Most legumes have fairly small genomes, and quite a few of them have now been sequenced to a high quality. Grass pea, however, which belongs to the same subgroup as peas and lentils, has a much larger genome. It's twice as large as the human genome. But sequencing technology has made amazing progress in the last few years. So together with Earlham and the University of Nottingham, we've been able to sequence and assemble this massive genome. With this data in hand, let's look at the biochemical pathway of how ODAP is made in the plant. We know a fair amount about the chemical steps in this pathway already. And we know the enzymes that do some of these earlier steps. But we also know that those same enzymes are involved in other metabolic processes that are important to the plant. So the best target is probably the enzyme for this final step, the one that makes beta-ODAP. From the chemistry of this reaction, we could have a decent guess as to what class of enzyme this is most likely to be. Looking at the sequences of all the enzymes of that class in grass pea and comparing them to the sequences in legumes that do not produce ODAP, I was able to narrow it down to a few candidates. By expressing these genes in a model plant that is related to tobacco and in bacteria, we were able to prove that one of these enzymes indeed produces beta-ODAP. So now we have a target gene. The most direct way to knock this out is to use gene editing with the CRISPR-Cas9 system, which we are doing with our partners at QUT in Australia. The particular application they are using is called SDN1. Briefly, this works by transforming the CRISPR gene cassette into the grass pea plant so that it inserts itself into the genome. Then the enzyme produced by the Cas9 gene makes a cut at a specific place in the target gene, elsewhere in the genome. As the plant tries to repair this cut, it sometimes makes a mistake, and that breaks the target gene. Then we wait for the plant to reproduce and to produce seed. We select from the offspring any plants that have lost the CRISPR-Cas9 cassette, but carry 2 copies of the gene change in the target gene. That leaves us with plants that have been gene edited, but do not contain any foreign DNA anymore. And that raises a very serious question. Should plants that once had a GMO ancestor, but do not contain any foreign DNA sequence themselves, still fall under existing GMO regulation? Some countries, including the UK and most likely the EU, think yes, while several South American nations have decided that no, these plants are more like the mutagenized plants we talked about before and should not fall under GMO regulation. Nigeria and Kenya also seem to be moving into this direction. But many regulators are still making up their minds. So it's unclear whether not-for-profit varieties produced by this method could be grown freely in Ethiopia or Bangladesh. And even in countries where GM crops can be grown, there are tight regulations that require seed manufacturers to produce a lot of data to show any genetically modified crop they want to bring to market is not damaging to health or environment. And this is one of the reasons why most currently grown GMO crops have been produced by very large seed companies that are able to shoulder this huge upfront cost. So in parallel to this work, we are also using a more laborious technique, which is called tilling, to produce mutants in the target gene without going through a GM intermediate. Like we did with the mutant screen earlier, tilling starts with a batch of seeds that have been treated with a chemical that causes mutations. After 2 generations, we then collect the seed from thousands of these mutagenized lines and store them. Each of them carries thousands of different random mutations all across their genomes. Separately, we extract DNA from the plants and store that. Then we can sequence our target gene in all of these samples to find any that carry mutations in the gene of interest. Then we go back to the seed library and pick out the corresponding seed packets that we can then use for further testing and breeding. This has a higher setup cost and a higher risk of failure than gene editing. In fact, we've had to solve this entire process twice already. But the great advantage is that material produced in this way would not be considered genetically modified anywhere, making it easier for breeders to produce varieties. We are now using both gene editing and tilling to generate mutants for the ODAP-producing enzymes. With both of these approaches, a lot of the work is in setting up the methods, especially in producing the mutant population for tilling. But once we have those, we can apply them for many other uses beyond reducing ODAP. By targeting the same genes that we identified in decades of breeding work in other legumes, we might be able to rebuild the same gains in grass pea in much less time. To recap, using the tools of crop science, we are now developing new traits for breeders to use and are measuring how these affect performance in the field. And together with our international collaborators, we are building tools that will make it easier to rapidly progress grass pea breeding, including genome and transcriptome data, and genetic maps and markers, which I did not have time to go into here. However, none of the work that we have done so far matters until these varieties reach the fields and plates of the people we are trying to benefit. Because having provably safe and high-yielding varieties is still only half the way. What keeps me up at night is not knowing whether this is enough for people to adopt these varieties. Will farmers believe that these new varieties are safe? Will this shift perceptions of grass bee as a food for the poor that is best avoided? And what other beliefs, practices, or economic factors could limit adoption? But these are questions that plant science does not have the tools to answer. To make my research matter to people like that farmer I met in Ethiopia, we need to understand the social context in which grass bee is produced, sold, and consumed. This has implications regarding agricultural policy, gender equity, consumer preferences, to name just a few. And this goes for my work on grass bee, as well as many other crop science projects. This speaks to an even greater question, which is whether the research that JSE and other bioscience institutes are doing is even on the right track. Research projects in basic plant science in the UK rarely start by listening to real-life farmers in low and middle-income countries. But without a good understanding of the complexities of agricultural development, there's a real risk that research outputs will end up being impractical or unappealing to the end users, or even harmful in unexpected ways. At worst, crop scientists can get locked into path dependency by trying to find an application for the latest exciting foundational insight rather than doing the work with the most potential on the ground. Norwich is the home to several excellent institutes working in biosciences. The John Innes Centre and Saintsville Laboratory, focusing on plant and microbial science, the Quadram Institute working in food research, and the Earlham Institute for Genomics. But it also has the University of East Anglia, with world-leading departments in international development as well as climate science. This is why I'm excited to be a part of the new NISD. The Institute will serve as a transdisciplinary hub, drawing in expertise from biology, social studies, economics, and climate science, all with the goal of better understanding how we can achieve more resilient and sustainable development around the world. Over time, we aim to develop research into many different aspects of sustainable development, but initially we will be focused on agriculture and food systems, as that is where the core strength of this partnership lies. This is a very new endeavor for both the Bioscience Institutes and the university. This hub will allow us to look at challenges in sustainable agriculture from two sides, through socioeconomic research to inform what is most useful, and through biosciences, which informs what is biologically achievable. I believe that there are 2 points of interface at which the potential for fruitful collaboration between these two disciplines is greatest. One is in setting the research agenda. Applied bioscience should not operate in a vacuum. We need to understand what will be the agricultural challenges of the future and then decide what we can address. What do farmers say they want improved? How will diet change affect future demand? And what future environmental stress will crops have to withstand? The second point of interface is studying how research outputs reach the field. How are they adopted by breeders and farmers? What impact do they have in terms of social and economic change, as well as climate adaptation and mitigation? These 2 points of interface do not exist in isolation, as experience from studying the impact of new technologies will feed back into directing future research. And both of these are necessary, but they operate on different time horizons. A key challenge is the long timeline for a new idea in crop science to turn into a useful tool in the field. And in some sense, this separates the 2 points of interface. Here's an example timeline for crop improvement. It might take 4 years to discover and describe a new trait in a crop species. Maybe this is somebody's PhD project. But it will take another 2 or 3 years to do proof of concept pre-breeding and develop genetic markers for that trait. Add another couple of years to transfer this technology to breeders in the relevant countries, who will then spend another 4 or 5 years breeding the trait into elite varieties suitable to their environments. Varietal registration will take another year. followed by 1 to 2 more years to produce enough seed to give or sell to farmers, and another 2 or 3 years of agricultural extension work. I've been working on grass pea for 8 years now, and we're still only at about step 3 of this. So in total, you're looking at about 15 to 20 years from idea to product. This is assuming there are no major roadblocks and that you start with an applied research question. Most of the work at JSC is more foundational than this, so the timeline will often be even longer. And this does not include time for developing ideas or attracting funding, or on the other hand, time for a new product to spread beyond its initial adopters and for users to become fully familiar with it. So if we want to see a research project all the way through, from defining a research question based on the development need on the ground, through all this crop science work, to evaluating the impact this new variety or technology has, we have to think in much longer timescales than normal grants and find ways to evaluate progress on the way. And we have to run multiple approaches in parallel because only some of them will pan out. Whatever you do, sometimes it turns out you're just not on the right road. Climate change will make this even more difficult. The world in 20 years will look different from now. The Arctic might be regularly sea ice free in summer. Dietary patterns will have changed, and populations will be larger in many already stressed regions. And hundreds of millions will have migrated to cities due to social and climatic changes. This is a world that none of us have ever seen. But this is the world that the crops and agriculture systems we develop now have to be suitable in. Let's return to this timeline. JSC only really works on the first 3 steps of this. I spent 3 years working in Kenya in order to be closer to the breeders, policymakers, and farmers that can see this research through to the field. There are many links like that along the research pipeline that we need to cultivate to maximize impact. And this is why I'm so keen to talk to you as people with such a broad range of expertise. Because it is only through effective international networks that we can do the difficult work of adapting, testing, and disseminating new agricultural technologies, and understanding the impact these technologies have on communities of farmers and consumers. One way we're starting to do this for grass pea is through the PhD work of our very own Matt Heaton. COVID and politics permitting, he will travel to Ethiopia this spring to study farmer communities in a key grass pea cultivating area. This will help us understand what trade-offs people are facing and how they would want to see grass pea improved, as well as what evidence and whose voices they would trust regarding the safety of new low-ODAP varieties. Having more insight into the systems by which farmers get grass pea seed they sow will help us better deliver beneficial varieties. Another project, which we're still looking for funding for, will investigate the history and current use of grass pea in the divided Bengal region. After being split into the Indian state of West Bengal and the nation of Bangladesh, this ethnically and linguistically connected region took different routes in regulating grass pea, with West Bengal even going so far as banning its sale and use as wages for agricultural laborers. To this day, both states use grass pea. But how did regulation change the use and views about this crop? How do our varieties perform, and what further improvements do farmers want to see in this region? How can we work with policymakers and farmers to support effective and sustainable use of this crop? So this is our social sciences approach to studying grass pea, which intersects with our work in crop research. Understanding how farmers use and perceive grass pea in its cultural context, policy environment, and history will make us better able to breed varieties that suit their needs. And insight into the acceptability and regulation of gene-edited crops and the grass pea seed systems will make sure that these are not left on the shelf. The Grass Pea project is one example of the kind of work NISD will be doing. Across our various projects, which you can read about on our new website, we are using transdisciplinary approaches to support sustainable agriculture. With world-class expertise in bioscience, international development, and climate research, Norwich is the perfect place for this institute. As we embark on this journey, we're thrilled to work with you, whether here in Norwich, at your institutions, or at that farmer's field in Ethiopia. I want to thank the many people who've been involved in this work so far, especially our collaborators across the NRP, at Nottingham, at Becker Ilri in Kenya, at EIR in Ethiopia, at ICADA in Ethiopia, Morocco and Lebanon, and UT in Australia. And thank you all for listening.
Thank you so much, Peter, for a wonderful presentation and also for keeping on time. I can see that there are several questions already on the chat box. Also, a couple of people had their hands raised. So while I request Natasha to maybe...
Oops, the sound has gone, Nitya. I don't hear you. Yeah. You were going to request that I pose these questions to both of you. Is that right?
Yes. Can you hear me, Natasha?
Yes, yes, that's fine.
Sorry.
Great.
Sorry. There's also a couple of people who had their hands raised. I was wondering that since we do have about 20 minutes time for questions and discussion, if we can request the people who've actually asked the questions to speak out their questions, it would be quite nice to get their perspective too.
Okay, I'm happy to moderate on that. Well, the first question was actually from Olusegun, which was around the toxicity of the grass and the seed, but I believe he's left the session. But related to that actually is Dominic Glover's question. So would you like to ask your question directly, Dominic, if you're with us? After that, we have a question from Jordan around the crop breeding and genetic engineering.
Yes, hello, can you hear me?
Yes, we can hear you.
Okay, so hello, first of all, great presentation, great project. So my question revolved around the fact that I'm sure that as most scientists in the plant science field have seen, a lot of great science basically comes undone upon impact with basically the legal systems or public opinion. And in several conferences I've been present in, including some with members of the organic food industry who are generally quite negatively opinionated when it comes to GM, the consensus at the end is that yes, there is a potential for improvement, but we don't like the concentration of breeder rights and the application of intellectual property rights to essentially the crops that feed us. And I was wondering if your institute will be using, well, whether it will include legal experts and whether you will be looking at the legal system surrounding GM, and whether you will be working towards a new paradigm essentially in breeding intellectual properties. So yeah, that was my question.
So Peter, would you like to, or do you want me to take that question?
Yeah, I'm happy to take it. Yes, you're speaking to about a very important issue here that affects many of the ways in which we can improve crops. I want to say that there's a huge amount of crop improvement that has nothing to do with GM technologies. There's a lot of work that goes on at NPI as well that doesn't use GM technologies, and that's all very useful stuff as well. But as far as GM goes, and as far as gene editing goes, and I've talked about some of the difficulties in the overlap between those two terms, that's a discussion that's very much going on in different countries at different pace at the moment. And we're seeing different regulatory paradigms coming out from different regulators at the moment. So this is a conversation where we will be very active, and where through our insight from both the policy conversation and from the plant science side, we will be able to add quite a lot of nuance. But we are very keen not to be seen as uncritically endorsing GM technologies or uncritically rejecting them either. So we want to see what we can do that will help people, and that might require GM technologies, but where it doesn't, we are very, very keen on doing the work that will get impact the fastest. And for example, with grass pea, that might include using a technique like tilling. That doesn't allow us to do everything we might want to do, but it will get us a crop that doesn't have that label attached to it. With regard to breeders' rights, that's an issue that also goes beyond GM, because even crops that are not using GM technologies still have breeders' rights attached to them. And that is an issue more to do with seed systems and who produces seeds, what quality of seed is available to farmers, and how that impacts the formal and informal seed systems on the ground. And that is something that we'll do quite a lot of research in. Again, that's not limited to GM technologies. It's understanding how seed reaches farmers and how we can build better networks so that countries have sovereignty over the seed that they use, which is relevant to their food production, but also can access the best seed.
Thank you very much. Just trying to sort out about the mics that aren't working and who is speaking. So related to that question, let me ask Dominic's question, which is around the toxins. And he was asking about, is the level of the beta-ODAP in the toxins in the grass pea affected by the processing and cooking methods? And that relates to an earlier question as well. And how have people who eat grass pea managed this issue till now?
Right. A very important point here is that people do know about, well aware of this issue and have developed techniques to process grass pea that reduces that risk. Some of those techniques are very well suited. If you steep the seed in water over an extended period of time, a lot of the toxin will leach out. If you then discard that water, and proceed to boil with new water, then you can get rid of quite a lot of the toxin, but not all of it. You can also partially detoxify it, but the toxin is fairly heat stable, so it's difficult to get rid of it entirely. However, there are other ways in which people process grass pea, where they roast dry seed or they eat the immature pods, where all of the toxin remains in what people eat. So there are some ways that people are using to reduce it, but you can't get rid of the toxin entirely that way.
Thank you very much. So we have two more questions. One was from Saskia Hervey, which is a broader question, and now I'll come to Tony, who's been patiently waiting with his hand up electronically. Saskia, I just want to read out your question. If you want to say anything more about it, say so after the response. But Saskia was asking really to Nitya around what are the plans for the NISD for public and policy engagement, and suggesting that this grass pea research could be potentially a really interesting case study for DEFRA to use around gene editing.
Nitya, do you want to take that question?
Yeah, I can. Just to say that thanks very much, Saskia, for this information about the DEFRA consultation, and we'll definitely be in touch with you. I think today is our launch. I think clearly for the next couple of months, we do have a plan, which we're still developing, in the process of developing, for public and policy engagement. I think one of the things that we are already got started with today's seminar is to have a regular sort of NISD thought leader seminar series, where we raise issues like this for discussion and get different viewpoints, different perspectives on board. Because I think it's quite important to discuss some of these issues which happen to be quite polarized in the global debate, but really to look at the pros and cons of some of these issues and to sort of unpack what is involved and what are the implications for different groups of people and for different stakeholders. So I think an important part of our work is in terms of public and policy engagement, we have the website. We are going to have a regular seminar series in terms of public engagement. We hope in the course of time, as our projects proceed and progress, to bring out shorter briefing papers and so on. But I think any opportunities like the one you've suggested would be really critical and very welcome for us to engage with. So thank you very much for that.
Thank you. Tony Allen, you have your hand up. Would you like to ask your question verbally? Well, we can't hear you, Tony, if you are speaking. Oh, can hear something there. Is that Tony? No, we don't have a response from Tony. Tony, if you can get your, oh, we've still got a hand up there. You've unmuted your microphone. Are you able to join us? Can you speak?
No, I can't.
We can hear you now.
No, sorry.
Does that mean there's not a question, or you can't?
Not a question.
Not a question. Okay, no problem.
Thank you.
Thanks. So we have a couple of questions now coming in from Rachel Warren and Rob Grant. Rachel was asking about how is the university supporting this new institute financially? Are they creating new positions? Is there any external funding at the moment? And Rob was asking a question from an economist around, is there any chance of doing similar work, eliminating the toxicity in cassava? And I think that would be pointed towards Peter, that one, and the first one to Nitya.
So thanks, Rachel. Yes, I think we are very small. As Peter presented, we have a small core team of 5 of us, and Matt Heaton, who's 6th, he's a PhD student, but he's been contributing quite centrally to our work. So yes, we do have some support in terms of our own time, and basically lots of good advice and lots of good conversations from the university and the partner institutes. The financial assistance has really been so far from the John Innes Foundation for our 2 fellows, Natasha and Peter. And we hope that as we develop projects, we apply for funds and grants and build our partnerships, that we will be able to expand and extend our team. We are looking already to put in some proposals for PhD funding and PhD fellowships. So hopefully by the end of this year, I guess it will take us a couple of months to build up, but we are hoping that by the end of this year, we will have, yes, a stronger group and a bigger group of people working on it. But having said that, I think both the university dev and the Norwich Research Park institutions have been extremely generous in terms of their time and sort of support, which is non-financial support, really.
Thank you, Nitya. And then the other question was around cassava toxicity to Peter.
Yes, that's an interesting question, actually, because the disease that beta cassava causes, which is called konzo, has exactly the same symptoms as the neurolathorism that you get from grass pea, but the 2 toxins are completely different. So at some point, the pathways of those 2 diseases converge and they act on the same system in humans. That's not something that we've been looking at, but there is quite a lot of research into the cyanogenic glucosides in bitter cassava that cause the cyanide to be produced when you eat it. That tends to be easier to get rid of through the food processing. So there are varieties with reduced cyanogenic glucosides, and typically people are all right with just chopping those up, having them oxidize or having them steep for a long time, which then reduces those cyanogenic glucosides before you eat them.
Thank you. We have another question coming in from Floris here, which is around, she's saying that in this case, it seems to be that social sciences come in to look at this after the project was conceptualized. And are we going to be developing some projects which have farmer first, essentially farmers' aspirations and local context starting at the start of this process, and how would that happen? So that's for both of you.
Yes, in this case, a lot of we've been doing this work for quite a few years now, from a crop science point of view, and this is something that kind of is coming into the NISD now as a research project that's already ongoing. But this is what I was saying with those 2 points of interface. We want to look into both how we can bring things into adoption that are coming out of research, but we also want to make sure that we direct research in the right way and use the insights from social science going into the other direction. So as we start up NISD now, we all come with research projects that we've already done, that are where we have expertise, where we have links on the ground, and we're building those links across the disciplines to now do research together, where we can make sure that the crop science is responsive to needs on the ground.
And Floris, just to quickly comment to that, I think as Peter showed towards the end, one of the slides that we've developed in Eastern India, Bangladesh and West Bengal is actually a ground level thing, because the grass pea was something which was indigenous to the area, which was consumed by indigenous people, very poor, but it started having the health consequences. So for instance, the legal issue comes in there too, apart from the historical one that the government of India had banned in the 70s. The trade of grass pea. It is still indigenous to the area and consumed, but of course it can't be traded or marketed, or is not marketable anymore because of its illegality. So I think this was a question that came up independently from the ground, from people saying that, okay, these are the problems, this is a great source of protein with climate change and so on, and what do we do? And then I think we discovered within DEV and from our NGO partners and communities on the ground that this was already an area of work which was going on in the center. So I think trying to build these bridges and set up these conversations between communities, between farmers groups, to identify what are their needs, what are their problems, and also make the link with the crop scientists. So I guess that's where we see actually something novel in NISD's approach, that the starting point may be from whichever end. But we hope that a lot of questions will actually start from the ground if it has to be more solutions focused, because I think addressing this whole problem of sustainable development and global food insecurity, we know that we can't wait forever. So we also want to do a bit of mix and match in terms of finding where the problems are, where the most nutritionally deprived, most food insecure populations are, and actually what are their issues, what are their crops, what are their problems, and then maybe try and build our work around that. And I think what Peter said around the farmers' rights and the breeders' rights is also, for instance, a country like India, it has a law now on recognizing farmers' rights, the rights of farmers as breeders, and there has been a lot of work with public sector institutions. And I think you made a point, somebody else had made a point about the international corporations and GM. So I think a lot of our work is also for the public good. So with public research bodies, whether it is global producing global public good, whether it is national research organizations or global research organizations. But to start off with, I think we are very mindful that this has to be a partnership for global public good rather than for private interest or private profit.
Thank you, Nitya. We just have one final question with a small addition as well from Oliver. He was asking around how the food safety of this new variety is going to be checked before promotion to farmers. And in addition to that, there is a question around, is this toxic for both human and animals, or just for humans, from Olusegun? That's for people.
The answer to that is quite related. It is toxic to lots of different animals, including humans, but the way that it affects different animals differs. So humans actually seem to be more able at catabolizing it, so breaking down this toxin, than some animals. So in chicks, for example, and in rats, you see direct effects when the animal eats a large amount or is injected with it, that you don't see in humans. In humans, it's only a chronic effect. And that has made it much more very difficult to get an animal model together that really shows the effects in humans. And that means we also don't have a known limit that we can say this is absolutely safe. We know that you really have to eat a huge amount of this toxin. So this is something like 50% of the calories that you're taking in have to be grass-B for 4 months. So this is really the cornerstone of your diet for people to start developing these symptoms. And that is with quite high ODAP varieties. So we are really quite sure that the low ODAP varieties that already exist are safe enough. But it's difficult to convince anyone that probably safe is good enough. So the best thing we could get would be a variety that doesn't have the toxin in at all. That would certainly be enough for any of the countries that already allow low ODAP varieties. But what we really need to make sure is that these levels remain under the legal limits that already exist under any environmental conditions, and that there is no environmental impact that can push it really high again.
I'm going to draw to a close, sorry, Nitya, and Oliver, we'll have to follow up with you after that into the final question that was just put to us. So I'm going to hand back to Nitya now. Thank you.
Thanks very much, Natasha, and thanks, Peter, for not just an excellent presentation for this virtual launch, but also for already generating a lot of interest, a lot of questions, and an excellent discussion. Thank you to everybody for joining this session and for your excellent engagement and questions. We definitely will keep in touch. I'm sorry that the last few we have not been able to address because that our time is up and we need to close. But I would again like to thank all the partners who have made NISD possible and a reality today. Please go to our website and watch the space there. We will be having very regular seminars like this, at least once a month, in order to keep this conversation going. And we'll be definitely following up separately with those of you who've asked questions or given suggestions in order to take this conversation forward. So thank you all very much for coming today. And we hope to keep in touch and we hope to build this together better. Thank you.